Okay, I also want to, before kind of getting into talking about the different viral vectors that are used for gene therapy, I want to draw your attention to the sort of timeline of assignments that are coming up at the bottom of this slide. So if you haven't already finished your draft of sections 4 and 5 of the research paper, you want to be working on that and trying to finish that up as soon as you can. Just a reminder to ask me for help when you need help. And then once you finish that, then you should go ahead and learn about gene therapy. For Tuesday, 4/14, there's a short gene therapy assignment due. That's the one I was referring to. I actually am suggesting you complete that assignment before you watch this PowerPoint. They kind of go hand in hand, okay? And then you can also watch the next PowerPoint for 4/14 that will tell you about microarrays. And there'll be a short assignment on microarrays that'll be due on Thursday, 4/16. And then I'll also be posting an example paper, just kind of explaining it, just so you get even a better idea of how microarrays are used. I've pushed back the deadline for the outline of part 6. So instead of Thursday, it's now due Sunday, 4/19. This is the part of the research paper where you're going to be proposing your own experiments. That assignment is posted and it's explained, but if you have any questions, please ask. And then the last thing on our list there is peer editing of parts 1 to 5. So hopefully by April 23rd, you'll have made all the revisions that you've gotten from me, and then you'll be able to swap papers with someone, and I'll assign those swap partners for some peer editing. Okay, so let's get back to gene therapy and let's talk about viral vectors. Now, what you're going to see on this slide and the following several slides are slides from the Flash section of that very nice gene therapy website. This is the part called Gene Therapy Tools of the Trade. And if you have Flash, you can actually just click through those slides on your own. It's interactive. Otherwise, if you don't have Flash, you can just look at these PowerPoint slides. Okay, so what we're looking at here are several different vectors that are used in gene therapy experiments. So a vector is just like a way to deliver a wild-type copy of the gene to the cells. So in this toolbox at the top, we have 4 types of viruses that are used as vectors. These are described on the next 4 slides. This will be helpful for question 9 of the 4/14 assignment. And at the bottom are 2 non-viral vectors, and those are described on slides 7 and 8, and this will be helpful for question 10. Okay, the first of the viruses is the retrovirus. So these are RNA viruses. The most famous is HIV that causes AIDS. You can see on each of these slides, I'm not going to read all the information, you can read it, but it tells you whether the virus can infect only dividing cells or whether it can infect both dividing and non-dividing cells. It also tells you in the target area if there is a maximum length of RNA or DNA that can be inserted into the virus. So you can see for retroviruses, the limitation would be 8,000 base pairs. If your gene of interest is larger than that, then retroviruses won't work. We can also look at the activation and the integration. And the integration is an interesting consideration, whether or not the DNA or RNA is going to integrate into the host cell genome, because if it does, it may actually interfere with some of those host cell genes if it's a random integration. And then side effects are also something to pay attention to in terms of immune responses and whether or not there's ways to sort of mitigate or help avoid those immune responses. So there's lots of considerations, and you'll be able to read about those for each of those, 6 different vectors. Okay, so retroviruses was the first virus. Adenovirus is the second virus. This is actually a double-stranded DNA genome virus. It can infect dividing and non-dividing cells. Maximum length of DNA, 7,500 base pairs. And this virus's DNA does not integrate into the host cell genome. In this case, what that means is the cell is actually going to kick out that DNA, and so the gene activation will be lost. And at the bottom, it tells you that adenoviruses are actually pretty well known to cause immune responses, so that's a concern when using adenovirus. The third virus, adeno-associated virus, it's called AAV. Unlike retroviruses and adenoviruses, AAV does not cause illness in humans, and the genome is single-stranded DNA. AAV requires a helper virus in order to replicate inside cells, and the maximum length of DNA you can insert into AAV is 5,000 base pairs. If you look in integration, you can see that AAV DNA will integrate into the host cell genome, and interestingly, it's not random. The majority of the time, it will be on chromosome 19. So that actually really reduces the possibility of interfering with host cell genes. And immune responses do not typically occur. HSV, herpes simplex virus, is the next viral vector. It is the cause of oral and genital herpes, and the genome is double-stranded DNA. The target is quite specific. HSV targets and infects cells of the nervous system only, and the maximum length of DNA you can insert into HSV is 20,000 base pairs. The HSV DNA will not integrate into the host cell genome, and it actually can stay in the cell nucleus for quite a long time, so it's more stable than the others, and it doesn't disrupt the function of other genes, so that's a benefit. However, HSVs can cause immune responses, so that's another concern. Okay, so we've now learned about the 4 viral vectors. This is the first of the 2 non-viral vectors. Liposomes are circular pieces of double-stranded DNA, which we also know as plasmids, and these can be packaged inside liposomes, which have lipids, lipid-based packets, basically. So if you add a liposome to a cell, these packets tend to fuse to the cell membranes, and the DNA can get transferred to the host cell. They're not specific for any cell type. However, the ability for the DNA to enter into the host cells is actually much less effective than viruses. There is no maximum length of DNA to package into a liposome. Liposomes are not going to generate an immune response, so that is something that you don't have to worry about, but they can sometimes be toxic. And at the bottom it tells you that liposomes are usually a better choice for an ex vivo gene therapy approach in contrast to in vivo. And you'll learn about the difference between those two with assignment 414. All right, the last vector is naked DNA, which is literally, like its name suggests, just double-stranded DNA in a circular mode, so it's a plasmid. It's not packaged in any way. It's not going to be specific for any cell type. It doesn't enter cells very effectively, but there's no maximum length of DNA that you could put into a vector. It's not going to integrate into the host cell genome, so there's not usually an immune response, nor is it usually toxic. And like liposomes, naked DNA is usually better suited for ex vivo gene therapy approaches. Okay, so those are your 6 different kinds of vectors that you could choose from when you're designing a gene therapy experiment. So let's go through some considerations about thinking about choosing a vector for cystic fibrosis. So we'll go through this case study, and again, if you have Flash, you can go to the URL below and click through the interactive case study. But if you don't have Flash, you can just click through slides 9 through 16 in this PowerPoint. Okay, so we're kind of picking back up from the case study we looked at earlier in the 331 PowerPoint. So after determining that cystic fibrosis is a good target for gene therapy, there's this consideration about how to deliver a normal copy of CFTR into a patient's airway cells. So the first choice is what vector to use, and that's what we're going to go through on the next few slides. The first question is about size. Every vector has an upper size limit for the inserted gene. The CFTR gene is 4,443 base pairs. So which vectors that are listed on the right-hand side of the slide can we exclude based on gene length? So to answer that question, you would want to go back to the info slides for each of the different vectors, and what you would see is that none of the above can be excluded because the CFTR gene is relatively small, so we're not going to have an issue with size. The second consideration, no virus can infect every type of cell, so we want to be mindful of what kind of cells we're targeting, and in this case it's airway cells. Are there any vectors we can exclude based on target cell type? And if you recall, the answer is going to be HSV because that can only target cells of the nervous system. So now that's kind of blacked out in our slide. The next question, what vector can you exclude based on its ability to only infect dividing cells? The answer is the retrovirus, so that's now blacked out at the top of the slide. So the retrovirus can only infect dividing cells. That's not going to work well for CFTR. Okay. The next question, what vector can you exclude based on its inability to enter cells very well? And you can already see the answer because at the very bottom, the liposome and naked DNA, those vectors are blacked out. They don't enter cells very well. So for this in vivo gene therapy approach, they won't be good choices. So this leaves us now with adenovirus and AAV. And the next question is going to be, which vector can we exclude based on its tendency to induce immune responses? And you can see that I've circled adenovirus. It's not going to get blacked out, but basically we're going to exclude adenovirus because it does have a pretty high tendency to induce immune responses. And that's going to leave us with AAV as the best choice to use as a vector for cystic fibrosis gene therapy. So the next steps would be to clone the CFTR gene into an AAV virus, and on the next slide I have a picture of how that would look. And then patients could potentially use an inhaler to deliver the modified AAV virus that contains the wild-type CFTR gene to their lungs. If you want to propose a gene therapy experiment in part 6 of your paper, make sure to consult the tip sheet for gene therapy experiments that's posted in the research paper folder. It'll help walk you through and make sure that you're including all the information that's needed. Okay, so let's take a look about how that picture of how it would look for CFTR to be cloned into the AAV virus. So this is actually just a generic cartoon showing gene therapy using adenovirus, but it would look very similar for any of the other viruses, including AAV. So at the top, we would have our new gene, that's the CFTR in our case, in pink, and you can see that that would actually get engineered to be part of the viral genome, and then it would get packaged into the virus. The virus would infect the target cells, so again, it would get delivered potentially to those airway cells of the cystic fibrosis patient. The virus would then enter into the cell. You could see it gets packaged into vesicles, and after a few steps, it's going to be able to bind to the nuclear membrane. And if we look on the far right of the slide, we can see the virus is then disassembled, and that means that the DNA can be released from the nucleus and be released from the virus and then enter through the nuclear pore into the nucleus. And once that DNA enters into the nucleus, it'll be able to be transcribed and translated. And that will hopefully provide functional CFTR protein to those airway cells of the cystic fibrosis patient. Okay, so now that we've learned quite